In our third step of glycolysis, fructose 6-phosphate was getting converted into fructose 1,6-bisphosphate. Here, phosphofructokinase 1 was the enzyme which was making this reaction possible, and it was using one ATP to take the phosphate of this ATP and put it on carbon number 1 of fructose, thereby making fructose 1,6-bisphosphate. Now, PFK1 is going to have two active sites. One of them, of course, is for the substrate, which is fructose 6-phosphate, and the other one is for ATP because it's using ATP to turn this substrate into the product. These are the two active sites of phosphofructokinase. Now, phosphofructokinase is highly regulated. It has 4 allosteric sites. When the energy level is very high, ATP is going to be high, and therefore ATP has an allosteric site on phosphofructokinase, which is going to inhibit it. At the meantime, citrate, an intermediate of the TCA cycle, is going to be increased when energy level is high. So it can come and tell phosphofructokinase to please stop glycolysis. When ATP is getting used and is getting depleted, it's going to be converted into AMP, which is adenosine monophosphate. And adenosine monophosphate's level will increase when ATP is decreasing. Therefore, it's only logical that AMP is going to be activating phosphofructokinase to tell the enzyme that we need more glycolysis to happen because we need energy. The last one, which is the confusing bit, is fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is going to be made through the action of another enzyme, which is called PFK2. Now, what happens in some tissues like the liver, we need glycolysis to go on even in states where energy is high. So what happens, our fructose 6-phosphate through the action of PFK2 is going to be converted into fructose 2,6-bisphosphate. So what happens is that our fructose 2,6-bisphosphate is getting made to come here and keep stimulating our PFK1 and to tell this enzyme that glycolysis needs to happen non-stop. So we are bypassing the inhibition that is going to be put through the high energy levels by ATP and by citrate. PFK2 by itself is also regulated by hormones, namely insulin and glucagon. Right after we have had a meal, the glucose level is high, insulin is going to come here and is going to dephosphorylate PFK2. And this dephosphorylation is going to tell PFK2 to please go ahead with this reaction. Why? Because we are going to need fructose 2,6-bisphosphate. Why? Because despite the energy level being high, we need PFK1 to work and to make more pyruvate. Conversely, when we are starving, someone who has not eaten for, let's say, 12 to 14 hours, glucagon level is going to increase. What will happen is that glucagon will come and phosphorylate PFK2, and it will tell PFK2 that please don't make fructose 2,6-bisphosphate. Why? Because we don't want PFK1 to work. Why we don't want PFK1 to work? Because we need to use the intermediates of glycolysis to make glucose. It needs to shift the biochemical pathways of liver into gluconeogenesis because the body is starving and we need to make glucose for the rest of the body. So glucagon will end up removing this strong activator of PFK1, so glycolysis can happen at a slower rate than when the body is well fed.